Good day everyone! Welcome to Activity 2. So today we are going to perform tests for antigen-antibody reactions. The following are the objectives for this activity. First, identify accurately hemolysis formed. Second, explain the theory behind hemolysis. Third, demonstrate agglutination reaction macroscopically. And lastly, describe the different grades of heme agglutination reaction. These are the following materials that will be needed in the procedure. First, we have toothpick or applicator sticks, test tubes, and clean glass slides. We will be also needing Nesco films to cover our test tubes, forceps, and pasture pipettes. Next, we have our micropipettes and pipette tips. The centrifuge will also be needed in spinning our test tubes. For the reagents and sample, we will be needing normal saline solution, distilled water, anti-D reagent, and our washed red blood cells, prepared 2 to 5% red cell suspension, and the patient's plasma. Now, how do we perform the slide method for the test for agglutination? To perform the test for slide agglutination, add 1 drop of anti-D reagent on the center of a clean glass slide. Add 1 drop of the washed RBC onto the center of the glass slide with the anti-D reagent. Using an applicator stick or a toothpick, mix well in a circular motion the blood sample and the reagent. Lift the slide and gently rotate or tilt back and forth and read agglutination result within 2 minutes. Agglutination on this slide can be seen as clumping of RBCs due to antigen and antibody interaction. For the negative control for the slide method, add 1 drop of NSS on the center of the slide using a pasture pipette. Add 1 drop of your washed RBC onto the center of the glass slide. Using an applicator stick or toothpick, mix well in circular motion. Lift the glass slide and gently rotate and read results within 2 minutes. The absence of clumping or agglutination means that there is no antigen-antibody interaction. Now we go to the tube method, which is another method for the test for agglutination. So how is this done? In performing the tube method for the test for agglutination, aspirate 100 microliters of the patient's plasma and transfer it into a test tube labeled as test. Next, add 50 microliters of your red cell suspension into the test tube labeled as test together with the plasma of the patient. Cover the tube with Nesco film and agitate to mix. For the positive control, add 2 drops of the anti-D reagent into the test tube labeled as PC, or positive control. Next, aspirate 50 microliters of the red cell suspension and add it into the same tube. Cover with Nesco film and agitate to mix. For the negative control, aspirate 100 microliters of the normal saline solution and transfer it into the tube labeled as NC. Next, aspirate 50 microliters of the red cell suspension and transfer it into the tube labeled as NC. Cover the tube with Nesco film and agitate to mix. The next step is to place the tubes inside the centrifuge, making sure that they are balanced. The tubes are subjected to a speed of 3,400 revolutions per minute for 15 seconds. After 15 seconds, remove the test tubes from the centrifuge using the forceps. Agitate the tube until the cell button is dislodged and observe for agglutination. Take note that a result is positive if there is agglutination observed. In this case, the agglutination grading is 4+ because we can observe one solid agglutinate. The other tubes are also agitated to dislodge the cell button and graded for the agglutination reaction. In this case, the agglutination grading is 0 since there are no clumping of red blood cells seen inside the tube. For the tube containing the test, it is also agitated, observed, and graded for agglutination. In this case, the grading is 4+ because we can see one solid agglutinate. Now we are done with the test for agglutination. Let's now move on to the test for hemolysis. In a test tube labeled as T, transfer 100 microliters of patient's plasma. Next, add 50 microliters of red cell suspension. Cover the tube with Nesco film and agitate to mix. For the tube labeled as negative control, aspirate 100 microliters of normal saline solution and add 50 microliters of red cell suspension. After which, cover the tube with Nesco film and agitate to mix. For the positive control, aspirate 100 microliters of distilled water and transfer it into the tube labeled as PC. Next, aspirate 50 microliters of the red cell suspension and add it into the tube labeled as PC. Cover the tube with Nesco film and agitate to mix. The tubes are then placed inside a centrifuge for a spinning time of 15 seconds. After 15 seconds, the test tubes are slowly removed from the centrifuge so as not to disturb the cell button. The tubes are then interpreted for hemolysis. In the interpretation of hemolysis, the supernatant and the cell button should be properly observed. And that ends the demonstration on how to perform the tests for the different antigen-antibody reactions. So I hope you learned something and thank you for watching.